and I'd just like to begin today by acknowledging the traditional owners and custodians of the land on which we meet, the Wurundjeri people of the Kulin Nation, and pay my respects to their elders past, present, and the elders from other communities who may be here today. So today I present Michael Coffey. Michael came to us as an honours student and did exceptionally well, and is now shared by my lab and by the lab of Justin Body. And so what he's going to tell you today about is a story that has turned out really well, but was put the panic in us when we realised that two of the top labs in the world were also working on the same project. So there was a mad rush to finish this work off, and I'm glad to say that we ended up with the best publication. It was really nerve-wracking though. And so what Michael has really done is he's built upon the work that's occurred in Plasmodium over the last decade and translated some of that knowledge actually into Toxoplasma. And this is kind of unique because often it goes the other way around in our field where Toxo informs what happens in Plasmodium. So this has been really interesting in the project as well, and it's been a really nice collaboration between Justin and my labs. So without further ado, thanks, Michael.
Is this working? Okay, thanks for that introduction, Chris. So today I'll be talking about the parasite Toxoplasma gondii and how this parasite is a master regulator of the host immune response. So Toxoplasma, upon infection of an organism, needs to mediate a fine balance where it can establish an infection and not be cleared by the immune system, but at the same time make sure that it doesn't overcome the immune system early during infection and cause injury or death to the host. And this is an immunofluorescence image here where there's two kinds of cells. So these large cells here are human fibroblasts, and these are what we grow our parasites in tissue culture. And these smaller cells here, these jelly bean shapes, are the parasites. So what I've done here is I've added a substrate that's been taken up by all of these cells and fluoresces green. And you can see these fibroblasts here are fluorescing green. However, some of these fibroblasts, for example, here are fluorescing blue. And that's because they've been invaded by these parasites. And during this invasion process, the parasite has actually injected in immune modulating proteins into these host cells. And one of these has been tagged as a reporter construct to make this cell fluoresce blue. And this is just one protein out of many that Toxoplasma injects into the host cell. So this is an electron micrograph of Toxoplasma gondii here, and Toxoplasma is a eukaryotic organism. And this is important because it contains a nucleus, and surrounding this nucleus is an endomembrane system where parasite proteins are made. And before secretion or export into the host cell, these proteins must pass through this Golgi apparatus and into some of these parasite-specific organelles. Toxoplasma is an obligate intracellular parasite, and this means that it must invade host cells in order to obtain nutrients and replicate. And any significant time spent outside of host cells leads to death of the parasite. Toxoplasma is also quite unique in that it infects any nucleated cell of any warm-blooded organism tested to date, so all bird species and all mammal species, and almost any cell, except for red blood cells within these organisms. It's an apicomplexan parasite, and apicomplexan parasites are defined by having an apical complex here at the apex end of the parasite, and this is involved in the initial attachment and invasion of host cells. Also in this apicomplexan phylum is the other Plasmodium species, which are the causative agents of malaria, and I'll be referring to these quite a bit today. And finally, Toxoplasma has specialized secretory organelles, so I'll be talking about these mostly during my talk today as well. One of these is the micronemes, or MN, shown here at the apex of the parasite, and this is involved in initial host cell attachment. The rhoptry organelles, these large club-shaped organelles shown as RH here, and these are involved in both attachment and invasion, as well as the dense granules, and these are electron-dense organelles here. And these are involved in modulation of the host post-invasion. So the Toxoplasma life cycle begins and ends with the cat or any feline species. So an infected cat sheds infectious oocysts into the environment. So these are active parasites, or dormant parasites, sorry, that are surrounded by an cyst wall, and these can live in the environment for years. These are then ingested by any intermediate host, and following infection, these parasites are active, they cause an infection, and then they lodge in the central nervous system and in the muscle tissue as these dormant, again, bradyzoite or cyst forms of the parasite. And then the sexual life cycle of the parasite is completed following re-ingestion of this cat by the cat, generally through predation, and the cat will then reshed these oocysts. So Toxoplasma can be ingested by any grazing animal, and it's a particular problem in livestock animals, especially in sheep. It can lead to a lot of death, premature abortion of these sheep, blindness, and mental retardation. However, in a lot of animals, it mostly just causes this infection, and again, these tissue cysts lodge in the muscles of these animals. And humans can become infected following ingestion of these tissue cysts, mostly in raw or undercooked meat. And this is a real problem in Central and Southern America and France, where a lot of raw meat is consumed. Humans can also become infected through ingestion of contaminated fruits, vegetables, or water that also harbour these cysts, and again, they become infected this way. In humans, the Toxoplasma parasite is generally well controlled by the immune system. However, in a subset of humans, it can really cause a lot of disease. And this is mostly mediated by this parasite, which is, again, these bradyzoites that lodge in the muscles, but also in the central nervous system and mostly in the brain. And it's expected that up to 30% of individuals throughout the world are infected with this chronic form of infection. Toxoplasma can also cause disease in pregnant women, and when they contract this parasite for the first time, it can lead to disease of the infant. So any disease caused by Toxoplasma is called toxoplasmosis, and one kind is congenital toxoplasmosis. So this baby here has been born with hydrocephaly, or an enlargement of the brain. However, infection earlier during pregnancy can lead to spontaneous abortion and mental retardation and blindness. Ocular toxoplasmosis occurs when the parasites form plaques at the back of the eye. So in white here is an active infection by these parasites, and in black is a previous scar. So when the parasites invade host cells, they multiply from 2 to 4 to 6, and generally lyse the cell when there's about 16 or 32 parasites. They then invade neighbouring cells and do the same thing, and this lytic cycle is involved in the symptoms of disease. As the parasite also lodges within the brain tissue, this is not generally too much of an issue we think it's not too much of an issue within immunocompetent people. However, following loss of the immune system, so if people develop AIDS, or if people receive organ transplants and their immune system's suppressed, these parasites can reactivate and in some cases form lesions within the brain. And toxoplasmic encephalitis is actually a leading cause of death in AIDS patients. And new work's starting to uncover that Toxoplasma may have a role in mental disease. So some case studies have shown at the moment that if people have Toxoplasma present within their brains, they may be more likely to develop schizophrenia as well as other mental illnesses. So this is a scanning electron micrograph here of a Toxoplasma parasite as it attaches to the host cell, and this host cell is present beneath the parasite. And in this zoomed in image here, you can see that the parasite is extruding its conoid as it attaches to the host cell. And this is involved in the initial attachment prior to this invasion step here, as the parasite actively pulls itself into the host cell using its actin-myosin motor. And this is a cross-sectional transmission electron micrograph here, essentially showing the same thing as this image, but you can see inside the parasite here. So the parasite, as this jelly bean shape, is halfway through entering the host cell, and it does this through entering by this moving junction. And you'll see in white around the top of the parasite here, this is actually the vacuole, the parasite vacuole that it makes as it enters the host cell, and the parasite lives within this vacuole, and this physically separates the parasite from the innate components of the host cell, including TLR receptors. As the parasite is invading, it's already released the contents of its micronemes, as I mentioned, which are involved in this initial attachment. And it's now releasing the contents of its rhopteres, these large club-shaped organelles, and these are involved in both invasion and the formation of this vacuole. And during this step, as the parasite's invading the host cell, it's already releasing host-modulating proteins. So host modulation is carried out by all parasites, and the success of these parasites rests on their ability to be able to successfully modulate the host. And parasites that have been attenuated and that can't modulate the host are generally cleared quite quickly by infection, by the immune system. So Toxoplasma is one of the most successful human pathogens. As I mentioned, it affects around 30% of humans. And we wanted to know, how does this parasite modify the host cell and also subvert the immune system? So these are some of the phenotypes that I'll be talking about today that Toxoplasma imparts on the host. The first is that it specifically phosphorylates STAT1, 3, 5, and 6 to skew the immune system early during infection. And then later in infection, it contributes to cell cycle arrest, the induction of c-Myc, and secretion of pro-inflammatory cytokines. Also, it recruits host mitochondria to the surface of this parasite vacuole, and it forms this unusual nanotubular network. So over the last decade, several Toxoplasma proteins have been identified that modulate the immune system. And the first ones of these identified were actually injected via the rhopteres during this invasion process. And not all parasite strains are able to effectively modulate the immune system to the same degree. And these were all discovered by quantitative trait loci mapping of crossing virulent versus avirulent strains and passing them through cats. So the first one of these proteins identified was ROP18. So ROP18 is injected during this invasion process, and you can see that the vacuole is starting to form here. And ROP18 coats the vacuole, and it phosphorylates and inactivates parts of the immune system called immunity-related GTPases, which would otherwise lyse the vacuole and release the parasite. Another one of these proteins is ROP16. So this is an immunofluorescence assay here, and I'm just going to take a bit of time to explain this because I'm going to be showing a lot of these today. So on this left-hand side here is a phase image, and you can see that there are 3 host cells present as they're slightly darker than the background. Each of these host cells contains a nucleus, and this is shown in blue here by a DAPI stain. So in any of my images today, whenever there's a blue image, it's DNA, or generally the host nucleus. And infecting these cells, you can see, are these small jelly bean shapes here, and these are the Toxoplasma parasites. So in general, these cells have around 10 parasites each. And shown in green here is a protein called ROP16. And ROP16 is another one of these ROP2 proteins that's injected during invasion. But this one has a nuclear localization sequence and specifically travels to the host nucleus. And in the host nucleus, you can see here in green, it phosphorylates STAT3 as well as STAT1, 5, and 6. And it's thought to do this to mediate a pleiotropic downstream effect that skews the host immune system away from clearing the parasite early during infection. So we've known for at least 10 years now that STAT1, 3, 5, and 6 are modulated through this ROP2 pathway, but how these other host cell processes were modified was not yet known. So this established the dogma, however, that host cell modulation by Toxoplasma occurs via the ROP2s during invasion. And this significantly differs to Plasmodium species, where most of the host-modulating proteins from the malaria parasite are exported post-invasion. However, as I've said, many of the host cell modulations from Toxoplasma could not be explained by ROP2 proteins alone. And recently, the dense granules, or those large electron-dense organelles that I've mentioned, are present throughout the parasite here as these dark spheres, have been implicated in protein export into the host cell. So the dense granules contents are constitutively secreted, and many of these Toxoplasma-induced changes are ongoing, so these are good candidates. And this is another immunofluorescence assay here, just showing ROP16HA. So again, ROP16 is just injected during this invasion process, and you'll see that, well, it's quite hard to see at the moment, but between 1, 2, and 6 hours post-infection, you can't really see any of this ROP16 in the nucleus, and it actually peaks there around 4 hours post-infection. So this wasn't really able to explain some of the changes that we see throughout infection. This is in contrast to the first exported GRA protein, or dense granule protein 16, which actually accumulates in the host cell throughout infection and increases in the host cell nucleus. So this first protein is shown to be exported post-invasion from the dense granules as GRA16. So in this phase image here, you can see the parasites are quite dark, and they're making this GRA16 tagged with a flag tag here. And you can see that GRA16 is present in and around these parasites, as well as co-localizing with this host nucleus stain here. If you pull out GRA16 from the host nucleus and look at it by Western blot, it specifically interacts and binds to this horse protein, or herpes-associated ubiquitin-specific protease, or USP7, which is involved in regulation of P53, as well as binding to several components of the protein phosphatase 2A complex. And it's thought that this altogether mediates cell cycle arrest by the parasite. So when the parasite invades, it doesn't want the host cells to replicate and divide, and it wants to arrest them in that state so that it can replicate it on its own. And what was really interesting about this GRA16 protein when we were looking at it for clues as to how it's exported was that it contains these 5 amino acids near the N-terminus of the protein, just downstream of this signal peptide that allows it entry into the endoplasmic reticulum. And this was interesting to us because it's very similar to an export motif in Plasmodium that was actually defined at WEHI in Alan Cowman's lab around 10 years ago. So these are normal erythrocytes, or red blood cells, as they pass through the bloodstream, and you can see that they have this biconcave disc-like shape. So red blood cells are what Plasmodium falciparum infects when it's in the host. And red blood cells don't contain a nucleus, they don't contain parasites, they don't contain protein secretory mechanisms, and they're quite devoid of nutrients. And so to infect these cells, the Plasmodium parasite needs to dramatically renovate them. And to do this, it exports over 460 proteins into the host cell throughout the life cycle. So this is a Drew Berry image here, just showing what an infected erythrocyte looks like when the parasites are lysing out at the end of infection, and these are the parasites here. So you can see that this erythrocyte dramatically differs in its morphology compared to these uninfected blood cells here. And that's because many of the proteins that are exported by Plasmodium are involved in remodeling and also adhering this red blood cell to the vascular endothelium, which stops the red blood cell traveling to the spleen where it could be cleared by the immune system. And I'm just going to say right now that around 12 years worth of really important malaria work, mostly done at WEHI, is going to be summarized in just a few slides here. So many exported proteins in Plasmodium that go into the host cell and modulate the host cell contain a Plasmodium export element, or PEXEL, and this was described by Matt Marty in Alan's lab in 2004. So the PEXEL motif is 5 amino acids commonly, and it's commonly defined as an arginine, any amino acid, a leucine, any amino acid, and then either an EQ or D in the 5th position. And this is a functional export motif. So if you take any N-terminus of an exported protein and fuse that to GFP, that GFP construct will be exported into the erythrocyte from the parasites, but not if it doesn't contain this motif. So this motif is exclusively found 15 to 30 amino acids from the signal peptidase cleavage site, and it's thought that this close proximity is absolutely essential for its function. So it was known that parasite proteins containing a signal peptide are co-translationally inserted into the endoplasmic reticulum, and then they have this PEXEL motif near the N-terminus of this protein, and somehow these proteins cross through the parasite cytoplasm, through the vacuole, and then enter the red blood cell. So again, skipping a few years of very important research, this motif is actually a protease recognition site, and it's cleaved at a conserved position here after the leucine. So this reveals the new N-terminus of the protein, which is then able to be trafficked into the red blood cell. Now in 2010, one of my supervisors, Justin Boddy, showed that the protease that actually cleaves this motif is called plasmepsin-5, or PMV. This processing step enables this new N-terminus of this protein to traffic into the vacuole, and then following that, it traffics through the Plasmodium translocon of exported proteins, or PTEX, which was described again in Melbourne, into the red blood cell. And this processing step by plasmepsin-5 in the endoplasmic reticulum is absolutely essential for licensing of this protein through the vacuolar membrane and into the red blood cell. And if you get cleavage revealing this new N-terminus of the protein by by signal peptidase in the endoplasmic reticulum, you don't get export. So plasmepsin-5 is absolutely essential for this step. And I just want to return now to Toxoplasma, where we know that the first exported protein post-invasion, GRAS16, is exported into the host cell, and it contains this RXLX, EQ, or D motif here. And we wanted to know whether this was essential for the export of this protein. So to do this, I tagged GRAS16 in Toxoplasma parasites with a HA tag on the end so that we could visualize this protein. When we look at it by immunoblot, we see that we get these 3 major species here. And when we looked at it just to make sure that it was still exported with this tag on, we see that the parasites shown here in red are making this protein, GRAS16 shown here in green, and they're exporting it into the host nucleus, as we'd expect. We then mutated this RRL to alanines, and it's shown that if you mutate the PEXEL in Plasmodium from RXL to AAA, you don't get export of this protein. And what we see is that this protein appears not to be cleaved anymore, and it appears to accumulate at this large molecular weight species here. So we think that potentially this protein is actually being processed at this point in the parasites. And what we also see is that this protein is no longer exported, and it's actually trapped within the parasites here, and it's no longer present in the host nucleus. So due to the similarity of this motif here with the Plasmodium export element, we named this motif the Toxoplasma export element, or TEXEL. So the plasmepsin-5 in Plasmodium, this endoplasmic aspartyl protease, cleaves the PEXEL, and it does this through its core aspartyl protease domain. And we then wanted to know, is there a TEXEL cleaving protease in Toxoplasma? So to find this out, we blasted this sequence of plasmepsin-5 against the Toxoplasma genome. And the top hit was for aspartyl protease 5, or ASP5, which is the focus of my talk today. So ASP5 contains several conserved features with plasmepsin-5, but most notably the same exact aspartyl protease domain. And within aspartyl proteases, there are 2 catalytic triads defined by an aspartic acid and a second aspartic acid. And these are what actually interact with substrates and are involved in hydrolysis and cleavage of these substrates. We tagged ASP5, and one of the first things that we noticed was that it actually co-localizes with a Golgi marker, and it's not present in the endoplasmic reticulum. We then pulled out ASP5 using anti-HA beads and incubated it with fluorogenic substrates. So these substrates are either contain the TEXEL of GRAS16 or this mutated version, which we know is not exported in the GRAS16 protein. And what we see is that ASP5 can cleave this GRAS16 motif, but not when it's mutated. Just to make sure that we weren't accidentally pulling out a protease that's not ASP5, but maybe is bound to ASP5 and mediating this function, we made a mutant version of aspartyl protease 5, or ASP5, where these catalytic acid residues here have been mutated to alanines, and this protein can't function as a protease. We see that it also goes to the Golgi, as we'd expect, so these residues are not important for its localization. But we see that when we use this protease, we don't get cleavage of GRAS16. So this showed to us specifically that ASP5 can cleave this motif in vitro. So using mass spectrometry of these cleaved products here, we know that ASP5 cleaves after the leucine at the same position as where plasmepsin 5 cleaves the PEXEL. And then Brad Sleeves went and made this peptidomimetic compound here, which mimics the GRAS16 RRL, and this contains an arginine, and a leucine. And this should fit into the active site of ASP5 and block its function. And Michelle really nicely described this with WEHI 842 and 916 on Monday. And these are the compounds that actively inhibit plasmepsin 5. And what we see is that when we incubate this substrate with ASP5, we get blockage or inhibition of ASP5's activity with an IC50 of 63 nanomolar. So this is very specific. And this showed to us this determined to us that ASP5 is cleaving this TEXEL motif in parasites. We then wanted to know what the substrate specificity of ASP5 is and whether it can cleave other motifs in the parasite. So using GRAS16 RRLAE as our control, we interrogated the P3 position here, the first arginine, and we see that mutation of this abolishes cleavage. The same is true at P2 and at P3. Sorry, at P2 and P1. So all of these first three amino acids appear to be required for cleavage by ASP5. We then mutated the P2' position, this alanine here, and we actually see an increase in cleavage, around threefold more. And we're not sure what the physiological relevance of this is in parasites yet, but we want to follow this up. And then just using our control compounds here, we see that again, when you make double or triple mutations, these aren't cleaved. So this is the TEXEL consensus that we came up with, and we think that ASP5 is preferentially cleaving proteins at RRLXX. So I just want to talk about what we know is the same and different between Plasmodium and Toxoplasma here. So we know that Plasmodium plasmepsin 5 is present in the ER, and we've shown that ASP5 is actually present in the Golgi. They both cleave this TEXEL or PEXEL motif at the same position, but they appear to have a different substrate consensus. So where Toxoplasma processes RRLXX, Plasmodium preferentially processes RXL, XEQ or D. So we think that this position is absolutely essential in Toxoplasma. We then wanted to know what the function of ASP5 is during infection. And to do this, we used CRISPR or Cas9 technology to attempt to knock out ASP5. And it's worth noting here that plasmepsin 5, the Plasmodium enzyme, cannot be knocked out to date using several different mechanisms, and it's thought to be essential. So I made two different constructs using CRISPR to target ASP5, and this is just one of them that targets ASP5 just upstream of the catalytic residues. In one of the clones that I sequenced, I saw there was an insertion of 2 Ts here, and this corresponds to a frame shift mutation where there's a premature stop codon, and we no longer get the functional ASP5 present. And just to make sure that what we were seeing was due to actual knocking out of ASP5 and not due to off-target effects by CRISPR, we did this with 2 constructs, and we made 2 independent ASP5 knockout lines. And I've used these for all of my experiments that I'm going to show today, just to make sure, just to validate that this is an ASP5 specific effect. So this is a plaque assay that I'm going to show now, and we grow our parasites, as I mentioned, in human fibroblasts. So following inoculation of fibroblasts into a dish, they'll grow to confluency, as they have here. We then leave them for 7 days, fix them, and then add a stain called crystal violet. And all of these cells take up this crystal violet stain, and you can see that they're all purple. When we add wild-type parasites to this and leave them for 7 days, they invade cells, replicate, and lyse those cells. And what we get are these large white spots here, or plaques. And these are zones of clearance where the parasite has invaded and replicated and lysed these cells. And therefore, there's no cells left here anymore. And this is kind of a crude growth assay that we can do. So when we use our ASP5 knockout parasites, what we see is that we still do get these plaques forming, but they're a lot smaller than these wild-type parasites here, indicating that ASP5 knockout parasites have some kind of growth defect. And when we complement back in ASP5, so we've got these parasite lines that are deficient with ASP5, and then we put in another functional version on top, we recomplement what we see here with the wild type, showing specifically that ASP5 is required for this phenotype. So we know that the parasites have a growth defect, and then we wanted to know whether they actually have a replication defect, because if these parasites can't replicate at the same rate as wild-type parasites, this would make sense. And what we see is that when we look at number of parasites per vacuole at 16 hours post-infection, we get comparable numbers of wild-type, knockout, and complemented parasites per vacuole. So this shows to us that ASP5 is not required for replication, and that it's potentially no longer mediating a virulence. And without this, it's no longer able to modulate the host or replicate as efficiently as it otherwise could. So we wanted to know how ASP5 acts on Gras16. And again, Gras16 is this exported protein, and when we mutate these amino acids here, we no longer get export. So again, just showing with normal Gras16, we get export of this protein, and we get these 3 molecular weight species on a Western blot. In our ASP5 knockout parasites, we no longer get this cleavage, and we get an accumulation of this larger molecular weight species. And furthermore, this protein is no longer exported into the host cell, and it's present entirely within the parasites. So we then wanted to know whether our ASP5 knockout parasites could be used as a tool to study other exported proteins in Toxoplasma. So I'm just going to return to this slide here, and as I've mentioned, ROP16 is involved in modulation of the STAT1, 3, 5, and 6. And we know that the cell cycle arrest phenotype induced by Toxoplasma is mediated by Gras16. We then wanted to know whether Toxoplasma can affect this c-Myc induction that it induces on host cells. So following invasion of host cells, Toxoplasma induces these host cells to express c-Myc. And this is present only, and c-Myc is a transcription factor. This is present only in infected cells and not in these other uninfected cells here that don't contain parasites. And interestingly, the level of c-Myc actually increases during the course of infection with Toxoplasma. And this indicated that it was not due to a rupture protein, which is injected during invasion and decreases in concentration, but potentially a dense granule protein mediates this phenotype. So in collaboration with the John Boothroyd's lab at Stanford University, and John gave a talk here during our centenary series last year and presented some of this preliminary work. He was able to show that a dense granule protein called MIR1 for Myc regulation 1 actually controls the activation of c-Myc. So MIR1 is a dense granule protein, and it contains a signal peptide to allow entry into the secretory pathway and therefore targeting to the dense granules. They were able to HA tag this protein, and they were able to specifically show that this protein is not actually exported into the host cell, but it co-localizes with a vacuolar marker. So this is the vacuole that the parasite resides within during infection. And this was really interesting to us, because we thought at the time that MIR1 would potentially be exported into the host cell and somehow mediating this upregulation of c-Myc. So I'll come back to this later and why this is important. We wanted to know whether our ASP5 knockout parasites are involved in this c-Myc phenotype. So when we don't infect host cells, just normal host cells, we don't really see any c-Myc. When we infect with our wild-type parasites, shown here in green, you can see that the nuclei that are expressing c-Myc are specifically infected by these parasites. When we use our knockout parasites, we get a massive depletion in this c-Myc and barely above uninfected. And then when we complement ASP5 back in, we again get this restoration of c-Myc induction in the host nucleus. This is just an immunoblot showing the same thing, where we get a massive induction in wild-type parasites infection and not in ASP5 knockouts. And what was really interesting when the Boothroyd lab shared this protein with us was that we noticed that around 2/3 of the way down the protein, it actually contains this RRLXX motif, which is exactly what we expect ASP5 to be cleaving. So we then pulled out ASP5 again and incubated it with these peptides containing this sequence, and we see that this MIR1 potential TEXEL motif is cleaved at the exact same efficiency as Gras16, but not when you make these corresponding single amino acid point mutations. We then looked at MIR1 using antibodies against the N-terminus in wild-type parasites, and we see that this is present at around a 70 kilodalton protein, which is the predicted molecular weight of the N-terminus of this protein if it was cleaved at this motif. And in our ASP5 knockout parasites, we get a 100 kilodalton protein, which is the entire full length of this protein. And then just to come full circle, using anti-HA antibodies at the C-terminus here, in wild-type parasites, we only get a 30 kilodalton protein showing up, and that's the approximate molecular weight of this fragment of MIR1. And then in our when we mutate this first arginine to an alanine, again, we get this accumulation of this larger molecular weight species. So this showed to us specifically that ASP5 is actually acting on this protein. And what was really interesting about this is that I mentioned earlier, the TEXEL motif is exclusively located 15 to 30 amino acids from the signal peptide in Plasmodium proteins. But we've been able to show here that ASP5 can cleave a TEXEL motif around 570 amino acids from this signal peptide cleavage site. And this may have something to do with the way that proteins are delivered to the Golgi, and that ASP5, when acting in the Golgi, can cleave this motif at any position. We then wanted to know what the effect of ASP5 is on the induction of pro-inflammatory cytokines by the host cell. And this was interesting. This protein, GRA24, was discovered not long after GRA16, but this protein does not contain any discernible TEXEL motif. So GRA24, again, is exported into the host cell from these parasites here and into the host nuclei. And once in the nuclei, it specifically upregulates the transcription factors EGR1 and CFOS. Pulling out GRA24 from the host nuclei shows that it specifically interacts with P38 alpha, or the MAP kinase protein 14, and it constitutively causes upregulation and phosphorylation of this protein. And it's actually shown that GRA24 induces the host cell to secrete pro-inflammatory cytokines that modulate the infection. And in somewhat attenuated parasites that don't express GRA24 anymore, so GRA16 has been deleted, the parasites actually kill mice and they overrun the immune system, whereas the wild-type parasites don't. So it appears that GRA24 is involved in making sure the parasites don't overrun the immune system. And GRA24, as I mentioned, does not contain this TEXEL motif. But just to make sure that we had the specificity site of ASP5 down pat, we pulled out some potential TEXEL motifs from this protein, but we see that none of them are cleaved with the same extent to GRA16. When we look at it by immunoblot, again, we see no difference in size, so no cleavage between wild-type or ASP5 knockout parasites. But we still wanted to know whether ASP5 was involved in the export of this protein into the host cell. So when we tag GRA24 in wild-type parasites, we see that it's exported from these parasites in red into the host cell nucleus. But in our ASP5 knockout parasites, this protein is completely trapped within and around the parasites and is no longer exported. And again, when we complement ASP5 back into these parasites, we get this restoration in export phenotype. So we know that ASP5 somehow is controlling the expression somehow is controlling the nuclear translocation of GRA24, and it's not acting on this protein itself, so it must be mediating this through unknown effectors, and we're really interested in following this up. One of the other really interesting phenotypes of Toxoplasma is that these parasites actively recruit host mitochondria from the host cell and decorate the vacuole that they reside within around. They recruit the mitochondria to this vacuole. So this is an image here of live parasites, and the host mitochondria are stained with mitotracker in red here. And you can see that these parasites, which contain this white vacuole around them, the parasites have recruited the host mitochondria. And this was described actually in the first immunoelectron microscopy of Toxoplasma in 1971. And they were able to show that only live parasites recruit these mitochondria and not dead or heat-killed parasites. So again, the Boothroyd lab were really interested in this phenotype, and they were able to show that a dense granule protein called MAF1 for mitochondrial association factor 1 resides at the interface of the parasite vacuole and the host cell and actively recruits these host mitochondria. And John again spoke about this during his talk last year. And he said that when they start pulling down MAF1 using anti-MAF1 antibodies, they also pull down some inner membrane proteins of the mitochondria, suggesting potentially that MAF1 reaches through and into the mitochondria. We wanted to know whether our ASP5 parasites were also deficient in this host mitochondrial recruitment phenotype. So Grant Jewson kindly supplied us with mouse embryonic fibroblasts, or MEFs, that contain this MTS GFP construct. So this is green fluorescent protein that's targeted to the host mitochondria through this mitochondrial targeting sequence. And we can see here that the mitochondria are located around this vacuole here, and it actually co-localizes to an extent with this MAF1 protein, which sits around the parasite vacuole. When we knock out ASP5, however, we get an aberrant phenotype where this MAF1 appears to be present just within the parasites, and maybe it's no longer trafficked correctly to the vacuole. And again, we don't really see any of this mitochondrial recruitment. When we put ASP5 back into these parasites, however, we get this restoration of vacuolar localization of MAF1, and then again, recruitment of this host mitochondria. So this was at 4 hours post-infection, and we wanted to know whether this is also true at around 24 hours post-infection. So I prepared some parasites for electron microscopy, and we sent these to David Ferguson at the University of Oxford. So he took this image here, and you can see that there are 4 parasites here, this one, this one, this one, and this one, all replicating within this vacuole. And all around this vacuole are these black arrowheads that show that the host mitochondria have actually been recruited and are sitting around the vacuole. This is in contrast to our ASP5 knockout parasites, however, where we only get a very small recruitment of these mitochondria and very little of the vacuole is coated with these. We quantitated this, and we see that in some cases up to around 45%, but generally around 25% of the host vacuole is actively coated in any cross-section with these mitochondria. And in our ASP5 knockout parasites, we get around 5%. And then again, just to see whether ASP5 specifically acts on MAF1, we see that we don't get any differential processing between wild-type and ASP5 knockout parasites, again indicating that somehow ASP5 is acting indirectly on MAF1. And then one of the last things we wanted to look at was the nanotubular network. This is a really interesting phenotype that the parasites produce themselves. This actually occurs within the vacuole here at around the base of the vacuole, and the parasites appear to make this nanotubular network, which looks like these long tube shapes here, and they're thought to aid nutrient acquisition and waste exchange by increasing surface area, but their exact function is not yet known. So this is in wild-type parasites, and we get quite a lot of this nanotubular network all around the parasites. In our ASP5 knockout parasites, though, we again don't really get any of this nanotubular network forming, and we only get some aberrant vesicles. So while the function of this isn't quite known yet, obviously Toxoplasma is doing it for a reason, and while the effectors that mediate this aren't yet known, we really want to follow this up. So we know that all of these phenotypes are affected in our Toxoplasma ASP5 knockout parasites, and the ones that we were specifically interested in are the ones that are actually exported into the host cell. So GRA16 and GRA24, and the upregulation of c-Myc, which we think would all affect transcription in the host cell. So I then performed RNA-seq on infected cells, and all of the RNA extraction and preparing of the cDNA library was done with the help of Stephen Wilcox at the Ian Potter Institute in the genomics division. And then all of the data was analysed and prepared for us by Alex Garnham and Gordon Smythe from bioinformatics. So what I'm going to show now is a heat map, and any genes in this heat map that are downregulated are shown in blue, any that are upregulated are shown in red, and any that are not regulated, specifically either way, are shown in white. And what we did was Alex prepared this graph showing the 100 most differentially regulated genes in host cells between uninfected host cells and wild-type infected host cells. So you can see that all of these genes at the top part of this graph here that are downregulated before infection are specifically upregulated following invasion and replication of these parasites. And the same is true where other genes that are already upregulated in normal host cells are downregulated during infection. And when we look at the host cells infected with the knockouts of ASP5, what we see is a dramatic reduction in the ability of the parasites to influence this host transcription, and they can't really go either way. So what we see is mostly an intermediate phenotype here, where these parasites can no longer mediate these dramatic changes on the host cell. So we then wanted to know, rather than looking at individual genes, what are some of the main biological processes that are affected during infection by these parasites? So these are some of the biological processes in the host cell that whose upregulation is dependent on ASP5. And we see that 4 out of 5 of these topmost biological regulated processes are involved in cell cycle. And this makes sense to us because we know that GRA16 is intrinsically involved in arrest of this cell cycle, and in our ASP5 knockout parasites, GRA16 is no longer exported into the host nucleus, and we don't think we get this cell cycle arrest. Also, looking at the other genes whose downregulation is dependent on ASP5, we see that 2 of these are actually involved in autophagy, and no current parasite processes or exported proteins are known to be involved in the autophagy pathway in host cells. So we think that maybe there'll be new effector proteins that we're yet to discover that are involved in this autophagy process. And we now have the ability to use ASP5 wild-type and knockout parasites as a tool using quantitative proteomics to find new effectors. So I just want to summarize now some of the changes that we know Toxoplasma induces on the host cell. So proteins are made here in the endoplasmic reticulum, and they're transported via vesicles into the Golgi apparatus, shown here in green. Within the Golgi apparatus is aspartyl protease 5, or ASP5, which I've been working on throughout my PhD. Proteins that are trafficked to the dense granules here must pass through the Golgi and may or may not be cleaved by ASP5, depending on their function. So we know that Gra16 passes through this pathway and accumulates in the host cell nucleus. The same is true, so in this pathway, Toxoplasma upregulates c-Myc, and it also exports Gra24. MAF1, the mitochondrial association factor 1, specifically recruits host mitochondria to the vacuole membrane, and we know that we also get induction of this nanotubular network. And finally, we know that all of these changes somehow mediate this large change in the transcriptional ability that the parasite's able to induce on the host cell. So we wanted to know then whether in ASP5 knockout parasites that appear to lack all of these functions, whether these parasites are virulent in mice. So as I mentioned, Toxoplasma can infect any warm-blooded organism, and we can take parasites that we've grown in tissue culture and put them directly into mice. So what we've done here is pooled groups of 6 C57 black 6 mice, and just as a control, we've injected these with just PBS alone. And what we see is that all of these mice survive this PBS injection, and none of them change significantly in body weight. When we inject these mice with PBS containing just 15 of these wild-type parasites, we get complete death of these mice by day 8. So these mice, once they either lose enough body weight down to around 85%, as laid out in our animal ethics guidelines, or when they start to look sick, we need to take them. So all of these mice were taken as they started to look sick. So we can see that by day 8, the wild-type mice have all been taken, and they started to lose body weight significantly by around day 5. When we put in our ASP5 knockout parasites, all of the mice survived the course of this experiment, and none of them really lost any body weight. If anything, they actually had this unusual aberrant gain of weight phenotype. And then again, to make sure that this was ASP5 specific, we put in our knockout/complemented line, and we see that again, all of these mice succumb to infection by around the same time. So just to make sure that our mice that were injected with knockout parasites did receive parasites, we did Western blots using their sera, and we see that they all have made antibodies against Toxoplasma and therefore were infected. And we then wanted to know whether, as they'd survived this initial infection, whether they were able to be, whether they could survive re-challenging with more parasites. So the mice that were initially just injected with PBS alone, when injected with now 50 wild-type parasites, and again, these wild-type parasites killed mice by around day 8 when we just used 15. So these PBS previously infected mice all died between day 8 and day 10, as we would expect, and they all began to lose body weight again by around day 5. In our ASP5 knockout mice that were injected previously, we see that we get a complete protection from this subsequent infection, and none of these mice lost any body weight throughout the course of infection. So this was work all done with the help of Seth Masters and Liana from Bioservices. So what's really interesting about this is that just one Toxoplasma parasite of this hypervirulent strain that I've been using is enough to kill a mouse. And even when we use 15 or 50 parasites, we don't get death. And in knockout of individual effectors alone, such as Gra16 or Gra24, show very, very mild delays in death, maybe 1 or 2 days, and never anything like this dramatic reduction and actual survival of infection. So we know that, well, it was previously thought, as I said, the dogma earlier, was that the Toxoplasma injects proteins into the host cell from the rhopteres during invasion. But we now know that there are 2 export pathways, one that occurs from the rhopteres during invasion, and another one via the dense granules that occurs throughout the course of infection. And trafficking and export of these dense granule proteins absolutely requires ASP5. So what I want to show now is just kind of like an accumulation of what we currently know in Toxoplasma. So this is the endoplasmic reticulum here, where proteins are delivered following translation by the ribosome. This is the parasite cytosol containing the Golgi apparatus and ASP5. This is the outside of the parasites here in the vacuole, and then up here is the host cell. So we know that TEXEL-containing proteins like Gra16 are made and present in the ER. These traffic to the Golgi apparatus, where they're cleaved by ASP5, and are then delivered into the vacuole via dense granules. And it's thought that somehow these proteins must cross the vacuole, and it's thought that they do this through potentially a translocon like PTEX in Plasmodium. However, this has not been defined in Toxoplasma yet. We also know that there are other dense granule proteins that don't contain this TEXEL motif. These are also delivered via the Golgi to the dense granules and then into the vacuole. So one of these is Gra24, which does not contain this TEXEL motif, yet somehow still crosses into the host cell. Another one is MAF1, which again does not contain a TEXEL, yet it somehow appears to localize into this membrane here and then mediate the recruitment of host mitochondria. And I mentioned earlier this MIR1 protein, this protein that the Boothroyd lab have discovered that's involved in regulation of c-Myc, and that this protein actually resides within the vacuole and not in the host cell here. And when they've blasted the sequence of this protein, they show that it actually contains homology to a bacterial translocase protein. And our current thought is that potentially MIR1 is needed to mediate the translocation of these proteins into the host cell. And then when we don't have ASP5 present here in the Golgi, MIR1 is no longer cleaved and can't exert this effect. So the next steps in my PhD are to identify the ASP5 substrates, and we're pretty sure that there'll be more of these yet to be discovered. And we're going to do this through quantitative proteomics. And we also want to characterize the translocon with the Boothroyd lab and determine whether MIR1 is actually involved in this pathway. So I'd just like to thank my co-supervisors, Chris Tonkin and Justin Boddy, as well as my committee, Alan, Jeff, and Stu. I'd like to thank everyone that was involved in this project from the Tonkin and Boddy labs. And there was, as Chris mentioned, there was really a mad rush to get this out as 2 other groups were working on it at the same time near the end of last year. So Brad made the compounds and did a lot of the peptide cleavage assay in vitro. Alex initially started the project by tagging ASP5, and Matt was involved in the original design of an ASP5 knockout construct. The Boothroyd lab, so John Boothroyd and everybody else here that were involved in helping us with the MIR1 aspect of the story. At WEHI, Alex Garnham and Gordon Smythe were involved in the RNA-seq data. Seth was involved in the virulence, and Grant supplied us with the mitochondrial targeted MEFs. At Oxford University, David Ferguson did all of the electron microscopy for us. And I haven't shown this data today, but at La Trobe University, Brian and Marta were involved in modelling ASP5 based on plasmepsin 5. And I'd just like to end there and take any questions. Thanks.
Thanks, Michael. Got any questions? Stu.
Really nice work, Michael. One of the features of the Plasmodium TEXEL is that glutamic acid, the +5, position 5.
Yeah.
And both of the TEXEL motifs you found had an E in that position as well.
Yeah.
So your alanine scanning says it's not important for cleavage.
Yeah.
But might it still not be important for targeting?
Yeah, absolutely. So we're not too sure at the moment at all what's involved in that, and we haven't interrogated that at all, but it's something that we've definitely noticed, and yeah, we'd like to follow that up further.
Did you say that the c-Myc was dysregulated in the ASP5 knockdown?
Yes, absolutely. Yeah. Or in the RNA-seq.
Yeah.
We haven't looked specifically for that. I hadn't really thought about that, no. We haven't looked, have we?
It's one of the top 100.
Oh.
Exactly whereabouts.
Oh yeah, it's actually, yeah, it's 52. Yeah, it is knockdown. Sorry.
Dunno.
You said in Plasmodium the function of plasmepsin 5 couldn't be bypassed by a second protease.
Sure, yep.
Could that happen in Toxo? Could signal peptidase touch the reported construct so that it reveals the mature TEXEL, and get exported?
Potentially, sure. We haven't thought about that at all. It could happen, because absolutely, we're not sure whether ASP5 cleavage is absolutely required or whether it can be cleaved by any other protease. Following on from what Stu said, if that glutamic acid is essential, as it is, as that P5 position is important in exporting Plasmodium, that would suggest probably not.
So you're saying that because it's found in the Golgi, not the ER, you might be able to make that bypass the requirement. Yeah, it's an interesting idea. Sharon.
In the in vivo experiments where you injected the parasites and you showed the death and of wild-type parasites and the ASP5 deficients do not, you didn't mention or you didn't show data on the parasitemia burden. So is there an increased number of parasites? Is there a greater number of cell death? What's the actual mechanism going there?
Yeah, so we didn't harvest the organs of any of these animals. To measure the parasitemia, it's quite difficult because Toxoplasma is a kind of systemic, but the main thing that we would have had to have done if we wanted to measure the parasitemia while they were alive was just do gavages in the peritoneal fluid, and that's not a great measure in Toxoplasma of the actual parasite burden. So it would be nice to go back again and actually have a look at that data if we weren't using, I guess, what we had to do here, which was more of death as an endpoint. So it'd be nice to go back and have a look at that.
Your TEXEL doesn't seem to be constrained to be close to the single peptide cleavage thing. And that one protein, I forgot the name of it now,
Yeah,
where you saw the N-terminus was 50 kD. Was that, but that's a bit of the active domain, I think. So,
Oh, I'm sorry?
B1. Where's the active domain in B1? He's asking.
So this was the protein that's cleaved around 500 amino acids further down. So MIR1 was only really discovered last year, and they're not too sure how exactly this protein functions. It doesn't appear to be an enzyme, so we're not too sure whether there are any active domains. It does appear that the C-terminus of the protein is actually present within the vacuole membrane, but yeah, we're not too sure how it's actually functioning yet. Just that when you knock out this protein, you no longer get c-MYC induction, as well as they've also shown now that you don't get export of GRA16 or GRA24 either.
Oh, Jason, one last question.
Great talk, mate. Do you know if anything else in ASP5, you showed the schematic, it looks like quite a lot larger than PMP. Do you know what the rest of the protein does?
Yeah, so we're not too sure at all with that at the moment. So it's quite a lot longer at the N-terminus of the protein, and it's actually predicted to have a signal sequence that may be cleaved off, or maybe not a signal sequence, but kind of that. The first 250, I think, amino acids of the protein aren't really predicted to do anything. The homology that it contains outside of the catalytic domain, and this was, we were able to model ASP5 by Brian and Marta were able to do this against plasmepsin-5, the crystal structure. And it appears that, yeah, ASP5 may have interacting domains with other proteins in the Golgi, but we're not too sure yet why it's so much larger.
Okay, so right on time. So join me in thanking Michael for a great seminar.